Good morning, everyone. Now I'm going to present you a efficient estimation of polygenic effects using a multivariate ridge regression, a study that I did in collaboration with my colleague David Xavier. So the presentation consists in a brief introduction and the rationale in statistical model, specifics about the coefficient estimation both at univariate and multivariate case, variance components both at univariate and multivariate case, and some simulation studies where we compare our method against the gold standard REML, and then some conclusions. Okay, let's get started with the introduction. So why should we even care about running multivariate models? We know that single trait models, they are already well known and well established for genomic prediction, and methods most deployed are such as GBLUP and BaseB. But we also know that phenotypes can come from multiple locations, years, and different traits. And we may choose to ignore the correlation among those traits running univariate models, or fit them through the multivariate framework and take advantage of their generic correlation. So basically, we know that whenever we model multiple environments and multiple traits, we capture complex G by E patterns. And as well as if we have multiple correlated traits, we can fit them together in order to achieve higher accuracy. Another thing that is novel these days is to assess the value of those traits derived from phenomic equipments and drones. And here I'm just giving an example of canopy coverage in soy. However, there's a big problem with multivariate models is that they are usually computationally prohibitive. So why would multivariate models be any better than simple univariate models? Well, whenever you have, this is a very simple case where a model is just your phenotype as a function of genetics and the residuals, and whenever you model those traits jointly, you choose to add to your model the covariance between traits in here notated as traits 1 and 2. So you get a better separation of the genetics and the residuals and how those two phenotypes can be associated in different levels. By not modeling them together, you lose all the connection you'd get otherwise through those covariance structures. And why should we do a ridge regression instead? Well, because whenever we fit a model using a ridge regression instead of the relationship matrix, we can more easily store the coefficients and use them for prediction. This type of framework is compatible to the multi-stage analysis, where you analyze in stages going from raw phenotypes to adjusted phenotypes, genetic merit across locations, and then the genomic breeding values. Another advantage is that ridge regression has very well-known properties, and we can get a lot of meaning out of those coefficients and a lot of, out of the genetic variance and residual variance. So they are Gaussian, they are additive, and they are equivalent to a GBLUP. Another advantage, computationally speaking, is that we do not need to build or invert the G matrix, which is not always positive definite. And it also, as I mentioned, provides some meaningful covariance components in the statistics that you can use for heritability, reliability, accuracy, correlations, selection indices, and correlated response selection. The statistical model of a ridge regression consists as follows. We usually have a summary of the genetic merit of the individuals. It's a vector. Mu represents the overall mean or the intercept. Z is the matrix of marker information, usually coded as 012 or -101 or centered. Beta are the regression coefficients, and E represents our vector of residuals. Under the multivariate framework, Y is a combination of vectors from different phenotypes. Mu is a vector with the overall means from the different phenotypes. Beta, likewise, is a combination of marker effect vectors. So are the residuals, and the Z matrix is a block diagonal where each block contains the genomic information from the different environments or different traits. The variance of the markers is described no longer as a single value but as a matrix. And the residuals here will be considered in a block diagonal, assuming that the residual covariance from two environments is 0. Because the residuals should not be correlated if they come from different environments. When we think about building the mixed model equation under the multivariate framework, we need to use Kronecker products to even build them. In this matrix, the whole left-hand side can be very cumbersome to compute. It's a very large matrix. It has all the combinations of Z prime Z across environments. It requires a lot of memory and CPU to compute. But that would be the classical way of solving a multivariate model in a ridge regression framework. So it's huge and it's hard to, it may require inversion to estimate the variance components, which have this solution right here. So in reality, it turns out that computing very large multivariate models is impossible. Well, at least if we keep thinking of them in the traditional sense. Alright, so let's start with solving the coefficients. Whenever we approach a whole genome regression like a ridge regression, we usually use the Gauss-Seidel method, which has as main idea updating one marker at a time. So instead of working with the whole left-hand side, we just solve one marker at a time, update the residuals, solve the next marker, update the residuals, solve the next marker. So it's a very simple system of equations. You update the marker, you update the residuals, you move to the next marker. This goes on and on until convergence, and here is just a depiction of how the marker effects would look like over the course of few iterations, and the goodness of fit increasing over time as well. In the multivariate framework, if we assume that the residuals are independent from trait to trait, we can rewrite the exact same algorithms, the Gauss-Seidel, in the following way. And the advantage of doing so is that it precludes you from having to build that entire Kronecker product left-hand side matrix. So you can solve the markers one at a time, solving a very minor system of equations that is just k by k, where k is the number of traits. So you update one marker and then subsequently update the residuals and move to the next marker, where again, that's just a small system of equations k by k. No Kronecker products are necessary, so you save a lot of memory by not having to build the left-hand side equation. So that's how the solution would look like. We basically have some operations that we compute once for the whole system of equations, which here are illustrated in dark green, so it's the cross product of the markers. Some are computed once every iteration, here in blue, which are the various components, both the genetic and the residual. And in orange, what is computed for every marker within every iteration. So basically it's the update of the marker effects, Z prime E for each trait, and then the update of the residuals. And what is in memory, we just have a minimal amount of things stored in memory, the marker, the coefficients, the residuals, the Z prime Z for each marker, so that's a K by M matrix, and the current version of the various components. One thing to notice is that if we choose to update the marker effects in random order, that can greatly speed up the convergence. So for instance, here I'm doing the update of the markers in random order. Within 120 iterations, I already reached convergence of minus 20, log 10 exponential minus 20. Whereas if I do the ordered genomic Gauss-Seidel, it took me almost 50,000 iterations to reach 10 exponential minus 8, which is the acceptable to call it convergence in most cases. Alright, so now moving on to the variance components. So we know that we should not undermine necessarily where variance components come from. We usually take for granted the whole history of the variance components, but here I just want to quickly go through the pathways of what is the gold standard and what we are going to be deploying in this study. So we can imagine that everything started with a simple ANOVA, was generalized by Henderson in 1953. Restricted likelihood, or RAML, came in 1962. And then we start to having a split between the iterative solutions and the exact likelihood path. So around 67 we got the exposure to maximum likelihood methods, which either went through the more quadratic expectation pathway, which is the mean Q and MIV Q, and they are both methods equivalent to what we know as RAML, proposed by Patterson and Thompson in 1971. The RAML has two or three main variations: the expectation maximization, derivation-free RAML, and more recently average information RAML from 1994. On the iterative path, we know that Thompson proposed a framework that is compatible to non-orthogonal matrices in 1969. And this pathway was further explored by Schaeffer and Van Reyden when, in the late 80s, when they wanted to solve for very large mixed model equations without having a computer power that we have nowadays, when at the time they used pedigree information instead of genomic information. So they created these fast variations that have satisfying variance components very close to what you'd get from RAML. So RAML, as a quadratic expectation, has this derivation: beta hat divided by the trace of centralized z prime z. The idea behind Schaeffer's or Thompson's pseudo-expectation method was to eliminate one of the v-1 matrices that yields just the centralized y prime z beta. So it's relatively simple and easy to compute, divided by the trace that you don't even have to update. We just compute it once. And then we have Van Reyden's method, which is a supposed improvement over Schaeffer's, where you leave, instead of removing v-1 altogether, you replace that by something very similar that he calls the d-1. So the method proposed by Van Reyden is called the Tilde-Hat, because instead of being the beta hat, he proposed calling this component of the equation b tilde, which is similar to what the v-1 would look like, but in a simplified way of computing. And all those methods, they can use the same residual variance as the same solution as the one proposed by Harville in 1977. And very easy to compute. It doesn't take much. Really, the problem lies on computing the variance components regarding the genetic variance. And one thing that we want to avoid at all costs is to build or invert V matrix, because that can be very large. Now, in the multivariate case, the solution coming from Schaeffer's and Van Reyden's are rather simple, right? So the only difference between Schaeffer's and Van Reyden is that Schaeffer would assume that that D matrix is just an identity. So all we have to do really, it does not necessarily involve any inversion. It's just computed from sums of squares and from the trace. So you don't even have to build the whole matrix, you just compute straight up the diagonals. So you do have some matrices in memory, but not nearly as many or as large matrices as you need for a RAML calculation. So the only thing that you have to compute is beforehand the z prime z. You compute it, again, I use the same colors to explain what you update every iteration and what you don't. And remembering that if you're using Schaeffer's method, this d is not really needed to compute. It's just the z prime z. So it's even less things that you have to keep in memory. So the beauty about using those methods is that you don't compute V, you don't compute the C matrix, which is the left-hand side, no left-hand side whatsoever, no determinants, and no dense inversions. So a more intuitive derivation of the Scheffer methods is presented here, where you can think as the genetic covariance between two traits is the same as the cross prediction between both traits. So you'd have the observed phenotypes of trait A multiplied by the A predicted from trait B, and do the same the other way around, where the phenotypes from B centralized multiplied by the predictions of B as they were predicted from this beta A from trait A. So it's basically the cross prediction divided by a normalizing factor, which is the sum of squares of the parameter space. And some key parameters that we derive from those multivariate statistics is the genetic variance that will be used for the calculation of the heritability of each trait, and likewise the genetic correlation between every pair of traits, here represented by K and K prime. Alright, but how good really are those methodologies that we are talking here? We know that Gauss-Seidel is an exact methodology, and the various components are an approximation. But if you put them together to solve intermittently the model, how well can we get if we compare them to RAML? And what's the accuracy, and so on and so forth. So to measure those comparisons, we are looking into three metrics. The accuracy of the breeding values as the correlation of the estimated breeding values and true breeding values. Recall that's a simulation, so we do have the true breeding values. We're going to check how good are the heritability and genetic correlations as they are estimated from the bias and precision. Bias is the difference between the estimator and the true value, and precision is the standard error by subtracting the estimated by the true value. And we are going to also look into the computational efficiency, which corresponds to how long it takes to fit such a model. And here's just an illustration of what bias and precision really mean. Precision is how close they are to the final estimator, and bias is how far they are from the true estimator. So things can be unbiased but precise. They can be unbiased and not precise. Biased because the average would be around here but not precise, or biased and precise, meaning you shot several times in the same spot but it's the wrong spot. Alright, so the first study is we're going to look into some width data. We have 600 individuals, 1300 markers, in a scenario where we have 10 environments, and that's a balanced scenario where all individuals have been observed in all environments. We're going to analyze RAML as the gold standard, the Pseudo-Expectation Gauss-Seidel, and the Tilde-Hat Gauss-Seidel that I'm going to refer to as pegs and tags. So the first thing that I looked at was the elapsed time after running it 100 times. And what you can see is that RAML takes much longer. So basically, to fit a RAML model, it took roughly 4 hours and 20 minutes, whereas the approximations in the multivariate scale took only 16 seconds. So it's a huge computational improvement. But if something is that fast, how does it provide the same quality of estimators? So that's what you're going to see next. First, let's look at the accuracy of the breeding values. And accuracy, of course, higher is better, and the highest value can possibly get is 1. So the largest discrepancy we got, so here we're going to be comparing this accuracy on the left in 3 levels of heritability and 3 levels of genetic correlation. So heritability low, medium, and high, genetic correlation low, medium, and high. So when the worst case or the most discrepant case we got was low heritability, low genetic correlation, where the difference between RAML and our approximations is 0.02. Overall, the methods were very similar. Multivariate methods were better than the univariate counterpart, and you can see that all the multivariate methods were better than the univariate. And as the heritability increases and as the heritability decreases and the genetic correlation increases, the discrepancy between univariate and multivariate models grew. But among themselves, all the multivariate models were satisfyingly similar. Now, what about the heritability? So the heritability was slightly underestimated by the PEGS method, the pseudo-expectation. The largest difference we saw between REMO and the approximations were when the heritability was high and the genetic correlation was low. And we believe that in part, PEGs have underestimated the heritability because it may have underestimated the genetic variance. Now, what about the precision of the heritability? That is the variance we find in each scenario. So precision is measured at standard error. So it's actually the lower the standard error, the better. And one thing that we notice here, that all methods were very similar, and they were all slightly better than the PEGS method. PEGs had a harder time to get higher precision when the heritability was medium or high. Alright, so now that we looked at heritability, now let's have a peek at the genetic correlations, which is the correlation among traits or among environments. So the methods were all generally unbiased, I would say. They were very close to zero, where PEGs was slightly biased upwards. The highest discrepancy we found was when the heritability is low and the genetic correlation is high. But this time, not because the approximations were bad, but because REMO underestimated the genetic correlations. So in this case, we do get a discrepancy, but not because of our methods, but because of REMO. Now, regarding the precision, again, we see that PEGs has a slightly lower precision than REMO and TEGs, the Tilde-Hat. But I would say that in this case, Tilde-Hat was actually better than REMO, and they are both better than PEGs. So we got a higher precision with the TEGs method. So you can more precisely estimate the genetic correlations in this case. So PEGs again had a hard time with the correlation, probably because it underestimates the genetic variance. So once the genetic variance is underestimated, then likewise the genetic correlations will be less precise. So in the overall summary of this study, we see that in accuracy, REMO and TEGs were the most satisfying. In terms of bias of the heritability, REMO was the best method. In terms of precision, TEGs, both univariate and multivariate, were the best methods. The genetic correlation was best estimated by TEGs, and the precision of the genetic correlations also best by TEGs, and worst by PEGs, which is the pseudo-expectation method. But one thing to notice is that the PEGs correlation was less precise than TEGs, but not statistically different than REMO in the balanced dataset. Now we're going to move to a second study that has similar settings, but now we are going to look what if we had 10 environments but no overlapping individuals in the same environment. So that means that we are going to allocate those 5,000 individuals in 500 individuals in each environment. So there will be no individual that has been observed in two environments. So each individual is observed only in a single environment. But because this data set has so many more individuals, we were not able to get REMO comparisons. That is, we tried to run REMO, but it took more than 2 days to fit a single model, so that was not conceivable for the whole simulation. Again, let's start from elapsed time. As the number of, so this one was what if we had an increasing number of environments. So for 10 environments, it took very few minutes, so less than a minute to fit PEGs and TEGs and the univariate. The time was still pretty conceivable up to 100 or 200 iterations, where it took less than 3 hours to fit the model. But if you go up to 500 environments, here for this particular study we we used all the individuals observed in all environments. So if we were to fit 500 environments and all 5,000 individuals, it took us roughly 14 hours. And if you are familiar with multivariate methods, you'd think that that's not unconceivable. Alright, so in terms of accuracy, both approximations were better than univariate, and one thing that you can notice is that PEGS is no longer lagging behind Tilde-Hat. So now they are very comparable. And once again, as the generic correlation is higher and the heritability is lower, multivariate methods were doing better than the univariate counterpart. So same trend as the WIT dataset. For the genetic correlation estimates, both PEGs and TAGs gave very comparable results in all scenarios, and they also provided the same precision, which increased according to the heritability and generic correlation. Now thinking of the heritability, once again we noticed that PEGs and TAGs are very comparable and they were better than the univariate counterpart. And in terms of precision, they were all very comparable. So all roughly the same, bias at 0.01 upwards, with the precision or standard error at 0.04. So here's the summary of both studies, where we see that REMO performs very poorly in terms of fitting time, and the fastest that we can get is that univariate from approximation. Accuracy is best with REMO and TAGs in the WIT dataset. REMO is the least biased method in terms of heritability, but in terms of generic correlation, TAGs was better both in bias and precision. But when we see larger unbalanced data sets where the individuals have not been observed in more than one environment, we notice that everything is about the same with PEGs and TAGs, and they are both slightly better than univariate methods. There are some other small limitations and considerations that I would like to point out. One is that we tested with a single fixed effect, which was the intercept. But you can compute with more covariates, but it can increase the computational burden of our proposed model. However, if you do a pre-adjustment, it should be fine. If you want to model correlated residuals, that won't be great for the computational aspects of it, because then you need to have the Kronecker product being computed. But it's okay in a multi-stage framework to assume the residuals are uncorrelated. What if you have kernels, like relationship matrices, or if your number of parameters is much larger than number of observations? So in these cases, the Gauss-Seidel can be costly. So a easy solution is to create a kernel and to eigen-decompose the kernel and solve the method using a ridge regression of eigenvectors, as described here. Another thing that must be pointed out is bending, because the variance covariance matrix of genetic covariances is not always inversible, and sometimes we have to deflate off diagonals until we make that inversible, and that can bias downwards the genetic correlations. Another thing to point out is that if my data is fully balanced, I can also run REMO efficiently by either running through canonical transformation or through a kernel diagonalization via eigendecomposition. So we point out that the time to compute REMO is very large, but it's only very large under regular settings. And also notice that it's only for balanced data, so there's no way we can get around unbalanced data with REMO. And last but not least, just a conclusion. So the remarks is that multivariate models are valuable because they were in all cases better than the univariate, but they have been historically computationally unfeasible, as you saw from the REMO time to fit a model. We proposed efficient estimation of coefficients using the randomized Gauss-Seidel and the variance components using Pseudo-Expectation or Tilde-Hat to enable multivariate ridge regression. And last but not least, we saw that TAGs and PEGs have some limitations, like the residuals cannot be correlated, and the model was very simplistic, just the intercept plus the markers. But it's a very good replacement for RAML in these simpler cases, and it can handle a very large number of traits and a very large number of markers, and a very large number of individuals. Thank you very much.